All right, so chapter 11, what we did last time is we went over the organization of the nervous system. And that is an important point. Until you understand how the nervous system is organized, is a little hard to move on to things like spinal nerves, cranial nerves, and especially the autonomic nervous system. So we went over the organization of the nervous system, and it is organized into the central nervous system and the peripheral nervous system. And the organization has to do with the function of the nervous system, which is to collect information, process information, and then put together a response. So the central nervous system is the integration center. So this is where the information is made sense of, and then a response is put together. This, as far as organs, this is the easy one, because the CNS has two organs, brain and spinal cord. So brain and spinal cord, that's CNS. And the job is complex, but it could be summarized as integration. Okay. And then we have the part of the nervous system that is going to collect information, send it to the CNS, and then it's going to take the response to the organs that are going to act it out, the effector organs. So we're going to have the sensory division or afferent division of the peripheral nervous system, which is sensory. And we're going to have also part of the PNS, the efferent or motor. So the PNS has two divisions, motor and sensory. So the two parts of the nervous system are CNS and PNS. And then PNS is divided into two parts, afferent and efferent PNS divisions, or sensory and motor PNS divisions. So the afferent division is the one that collects information. And the organs that collect information are going to be called receptors. And these are neurons. So these receptor neurons are going to collect information from the external world, like vision and sound and touch. So we call them exteroreceptors. From muscles and joints to give us information about the location of the body in space, and then we call them proprioceptors. These receptors will be found in muscles and joints. So the nerves that carry these information to the CNS are part of the afferent PNS division. And more specifically, they're going to be somatosensory division of the efferent PNS. So this is somatic sensory. So a nerve that carries visual information to the brain, it is a somatosensory nerve. It has a somatosensory function. It's part of the afferent PNS division. We also had interoreceptors, which carry information about what was going on inside the body, like changes in blood pressure, temperature, levels of oxygen, CO2, etc. And so those nerves were part of the visceral sensory division. Okay. So the somatic sensory or visceral sensory has to do with where the information is coming from. Okay. Now, these nerves that are carrying this information to the CNS, according to chapter 11, are going to be made of unipolar neurons. A few of them may be bipolar, but those are very, very few and far in between. Those are just the retina and the olfactory ones. So unipolar neurons are the ones that have the little body to one side and the one appendage going from one place to another. So sensory or afferent PNS division uses unipolar neurons. Again, bipolar also, but by far, nearly all of them are unipolar. So that's the afferent sensory PNS division. Let's go for the efferent, and the efferent is a division that carries commands to effector organs. And those are going to be not organs of the nervous system, they're going to be organs of other systems. But the nerves that carry these commands to these organs are part of the motor efferent division. So the effector organs can be skeletal muscle, and in this case, the nerves, the division will be the somatic motor. Okay. The organs, the effector organs can be cardiac muscle, smooth muscle, or glands. I don't know how to draw a gland. Let's just make something like this for a gland. And in this case, the division is going to be the visceral motor or autonomic nervous system, which acronym is ANS. And in this case, there are going to be two types of divisions of the ANS, which are going to be the sympathetic and the parasympathetic. The neurons for the motor division are all multipolar, because multipolar neurons are motor neurons, except for the association neurons, which are also multipolar, but they are association neurons, and they look different from the motor multipolar neurons. All right, so the ANS has two divisions, sympathetic and parasympathetic, and that is because these commands are going to organs over which we have no control. So we cannot will the organ to go or stop. This is an automatic process controlled by the ANS, by the central nervous system, the autonomic parts of the central nervous system, and the command is going out towards the organ via the ANS neurons. Okay, so because there's no control, we need two innervations. One set of nerves is going to make the organ go, the other one is going to make it stop. And that's why we have sympathetic and parasympathetic. The problem is, what makes the whole process confusing is that depending on the organ, the sympathetic division is going to make the effector go, and the other one will make it stop. And in another organ, the parasympathetic will make it go, the sympathetic will make it stop. So it has more to do with the type of response that that organ is involved in rather than the sympathetic or parasympathetic itself. So let me give you an example. For the heart, which is an organ that will respond in the case of a stressful situation. So in that case, sympathetic division makes the heart go, parasympathetic makes it stop, meaning stop, slow down. So sympathetic makes the heart rate increase, parasympathetic slows down the heart rate. By the same token, on the other hand, the GI tract, that is a set of organs that respond when we are under rest, relaxation, digestion. So in that case, parasympathetic will make those organs go, sympathetic will make them stop. So which is the acceleration pedal, which is the stop pedal, has to do with the kinds of response that will be elicited by the organ. Okay, so it's almost the same in a race car. Sympathetic is go, parasympathetic is stop. In a tractor trailer, or let's say, I guess, at the trailer, or a mobile home, the parasympathetic, something that goes from vacation and relaxation, I guess that's what I'm trying to think. Parasympathetic makes it go. Sympathetic is the accelerator pedal. Sympathetic is the stop pedal. Okay, so but we'll have a whole chapter on these, and that we're going to go over. But I wanted to put it forth early on. So this is chapter 11. You definitely have to understand the organization of the nervous system. Part of what we did in this chapter also was go over the cells, the types of cells, glial cells, neurons, make sure you understand the neuron, the parts of the neuron, etc. We also went over how the neuron works, and we said that in general neurons are going to respond to a stimulus. Typically we draw a multipolar neuron on these instances, which means that a multipolar neuron is going to be a motor neuron, and the stimulation for this neuron is going to come from another neuron. So we're not looking at a receptor neuron. We're looking at a neuron in which there is a pathway. It's a series of neurons on a pathway that are relaying information from one to the next. So let's say this neuron number 1 here is the one that is receiving the command from the CNS to go. Okay, so this neuron is going to create an action potential, which is going to release a neurotransmitter on the synapse between the two neurons. And when the second neuron receives the neurotransmitter, it will be depolarized by sodium ions that are going to go in, making the cell more positive from the resting state, which is negative. This is the graded potential, which takes these positive charges to the axon hillock, where they're going to be voltage-gated channels. Specifically, initially sodium channels, which are going to open when the value, the voltage of the area reaches a certain threshold value, which is about minus 55 millivolts. Remember, resting membrane potential is minus 70 millivolts. So positive charges coming in from the greater potential make the area more positive. When it reaches minus 55 millivolts, all the voltage-gated sodium channels open and the area is depolarized all the way to positive 30 millivolts. This was called depolarization. Definitely know what's happening on the curve. So there will be the depolarization, and then at plus 30 millivolts, voltage-gated potassium channels open, and now the cell will begin to lose potassium because voltage-gated channels are going to be open, and the cell will repolarize, overshoots to minus 90 millivolts. So it's going to become very negative, and then the sodium-potassium pump will bring it back to to the resting membrane potential. Okay. So the loss of potassium ions from inside the cell takes the cell all the way to minus 90 millivolts. So make sure you know what an action potential looks like. Make sure the steps of an action potential, what is depolarization, what is hyperpolarization. Okay. And finally, this is just a very quick overview. The last thing we went over were the groups of neurons, group A, group B, group C, and group A neurons were myelinated large neurons. So these were somatic sensory and somatic motor. And you should know what these terms mean. And you should know what kinds of receptors are involved and what kind of effectors are involved that have group A neurons. Then we have group B neurons, which were lightly myelinated, medium in size, and these were autonomic sensory and not autonomic, visceral sensory and autonomic motor. Okay, so intrareceptor neurons and ANS motor neurons. The speed of conduction was very fast for the group A neurons, and the means of conduction was saltatory. That's why it's so fast, is because this is a myelinated neuron. Group B, it is fast, but it's not as fast as group A. And again, saltatory, because if this is lightly myelinated, this is still myelinated. And then the slowest of neurons were group C, which were small neurons, slow neurons, non-myelinated, and these were ANS neurons also, actually, and visceral sensory. Okay, so remember what group A, B, and C are. Oh, and the conduction was continuous propagation because they're non-myelinated. All right, any questions of any of these? Okay, so let me go ahead and wrap it up with chapters 11, part C. And this looks at the synapse, which are the places where two neurons come together. So we're going to have a presynaptic neuron before the synapse and postsynaptic neuron after the synapse. So here is a synapse between neuron number 1 and neuron number 2. And you can see how neuron number 1 reaches the body of neuron number 2 and releases its neurotransmitter close to the body of the receiving neuron. The other example of another one of these synapses is when the neuron number 1 is synapsing with the dendrites of neuron number 2. So remember that the receiving regions of a neuron was the soma and the dendrites of the neuron. Those are the places where these synapses take place. The other types of connections that the book talks about, axosomatic, axoxonic, dendritic, dendrosomatic, these are not only less common types of connections, but they are found during development. So during fetal and especially fetal development. So they're not mature connections. Mature connections after we're born, axosomatic and axodendritic are the connections that we see in neurons. So these are chemical synapses, and at the connection, first of all, the two neurons never physically touch, even though in the pictures they look like they're touching. In reality, they're not. And there is a space into which a chemical, a neurotransmitter, is released. The axon terminal of the presynaptic neuron contains the synaptic vessels with the neurotransmitter, which will be released into the synaptic space. And the receptors for the neurotransmitter, are going to be found in the postsynaptic neuron, which is the one that is receiving the neurotransmitter. So we have here, this is the axon terminal of the presynaptic neuron. This is dendrite of postsynaptic neuron. So these are the vesicles with the neurotransmitter, which will be released into the synaptic space, and the receptors for the neurotransmitter are going to be found on the postsynaptic neuron. So make sure you keep that straight as to what is the presynaptic neuron's role, what is the postsynaptic neuron's role. We saw this last time on chapter 9, what causes the release of the neurotransmitters and influx of calcium into the axon of the presynaptic neuron. Okay. And the neurotransmitter diffuses and binds to a receptor located in the postsynaptic neuron. All right. The neurotransmitter must be released, travel down the synapse, and bind to the receptor. So between the time that the neurotransmitter is released and it binds to the receptor, there is a time that goes by, which is called a synaptic delay. So the time that it takes between the release of the neurotransmitter and a signal appearing in the postsynaptic neuron. That's the delay. All right, so there is the neurotransmitter binding to the receptor. The receptor is an ion channel, which opens upon the binding of the neurotransmitter, and ions are going to go in, sodium ions typically, not always. Potassium ions go out. We really did not talk about the potassium ions going out. We just focus on the sodium ions going in, making the membrane potential more positive and depolarizing the cell. Okay, so this is how we explain what happens when the neurotransmitter binds to the receptor. Now, coupled here, this may not be what always happens, but this is how we explained it initially as the event that happens. This is what we call the graded potential that is being created by the stimulus. In this case, the stimulus is the neurotransmitter. Now, this direct effect of the neurotransmitter on the receptor may not always be the way it works. Sometimes there is an indirect effect in which the binding of the neurotransmitter to the receptor causes a series of events that will open an ion channel and allow for the ions to go in. So the receptor is only that. It's just a receptor which initiates a series of events that will indirectly open ion channel. But again, just letting you know, this is not always the way it happens, that the receptor is the channel. Sometimes it's not the channel, it's just a receptor that through a series of indirect events causes the opening of a channel. All right, the neurotransmitter itself doesn't stay around for very long. It will either be degraded by an enzyme, as in the case of acetylcholine being degraded by acetylcholinesterase. It may just diffuse away, or it may be reuptake, brought back in the presynaptic neuron. So the graded potential is graded, meaning it can be strong or weak. And the strength of the graded potential has to do with how much neurotransmitter is being released, the type of neurotransmitter that's being released, and the type of receptor that the postsynaptic neuron has for that neurotransmitter. So ultimately, the receptor does play the main role, the main say-so as to what happens next. So it's not so much a feature of the neurotransmitter itself as it is a feature of the receptor. Okay, so we talked about these already. All right, so let's talk about the receptor. Depending on the receptor, the graded potential, so what happens next, could be an excitatory potential, meaning postsynaptic neuron will create an action potential, or it could be an inhibitory postsynaptic potential, in which the postsynaptic neuron will not create an action potential. It will be shut down. All right, so EPSP is an excitatory postsynaptic potential, and this is what we talked about on PowerPoints A and B. This is when the receptor opens and sodium goes into the cell, depolarizes the cell, and may create an action potential if enough sodium ions go in. This is what happens when the cell is at rest. Graded potential brings in sodium ions, and the sodium ions make the cell more positive, and this is happening at the axon hillock initially, and then moves on down the axon. It brings in enough positive charges to move the cell towards threshold and potentially create an action potential. Okay. Now, the receptor, on the other hand, could also be a potassium channel or a chlorine channel. Let's talk about the potassium channel. We're going to go ahead and ignore the chlorine channel, even though the effect will be exactly the same. If the receptor for the neurotransmitter is a potassium channel, so this is the cell, okay, and here's the receptor, and here comes the neurotransmitter secreted by the presynaptic neuron. If this is a potassium channel, when the channel opens, now potassium is going, which is inside the cell, is going to escape out. So the cell will begin to lose positive charges. And when that happens, the cell becomes more negative. So from minus 70 millivolts, the cell turns to even more negative values, let's say minus 80 millivolts. So the cell hyperpolarizes. So now we're going in the opposite direction. There will not be an action potential created. So if the receptor for the neurotransmitter is a potassium gate, potassium is lost by the cell, meaning positive charges are lost by the cell. If the receptor is a chlorine channel, chlorine is gained by the cell, meaning the cell will gain negative charges. Either way, the result is the same. The cell hyperpolarizes, becomes more negative. So instead of going towards threshold, now it's moving away from threshold. There will be no action potential possible. In these instances, the cell is inhibited. That's why it's called an inhibitory postsynaptic potential. Okay. Now, another principle here is that a single EPSP, one cell releasing neurotransmitter, is not going to create an action potential. There's not enough depolarization going on. What needs to happen is what we call summation, whether it be temporal summation or spatial summation. Temporal summation happens when a neuron releases neurotransmitter continuously onto a postsynaptic cell over a short period of time. So here it's giving you 2 releases of neurotransmitter over a short period of time that releases enough sodium ions to bring the cell to depolarization. That's temporal summation. The spatial summation is when there are 2 presynaptic neurons. These are the dendrites of the neuron, which release the neurotransmitter at the same time. So you have 2 firings by 2 different cells, bringing in enough positive charges to create an action potential. So that's a spatial summation. Okay. Ultimately, we are showing one neuron being synapsed by 1 or 2 neurons, and that's not realistic either. Most neurons synapse with hundreds, sometimes thousands of neurons. So this is extremely complex. Some of these neurons synapsing may be secreting neurotransmitters that create an EPSP. Others may be secreting neurotransmitters that create an IPSP. And essentially it is addition and summation of what is being released that determines what happens to the cell receiving all this input. Now, what you need to know from here is you should know what an EPSP is, and that an EPSP does make an action potential possible. What an IPSP and an IPSP does not going to make an AP possible. You should know what we mean by temporal summation. You should know what we mean by spatial summation. Okay, neurotransmitters themselves, most neurons make more than one type. There has been about 50 neurotransmitters or more identified. They are classified by chemical structure and by function. There are many different types. Ones we have talked about, we talked about acetylcholine in chapter 9. You should definitely know acetylcholine. This is the neurotransmitter released by somatic motor neurons. And also by some ANS motor neurons. This is the one that's degraded by acetylcholinesterase. Another group of neurotransmitters are the catecholamines. Dopamine and norepinephrine are the catecholamines. And norepinephrine is a really important neurotransmitter, which is going to be also a neurotransmitter of the autonomic nervous system, the visceral motor division. Indolamine, serotonin, and histamine, these are neurotransmitters that are extensively used by neurons of the GI tract, which is called the enteric nervous system. Endorphins, another type of neurotransmitters. So know the neurotransmitters mentioned. Also remember that the effect of the neurotransmitter is determined by the receptor in the postsynaptic neuron, not by the neurotransmitter itself. Acetylcholine is excitatory in skeletal muscles, inhibitory in cardiac muscle. So it's not the acetylcholine that makes it excitatory or inhibitory. It's the receptor for acetylcholine that makes it excitatory or inhibitory. So remember that this information being gathered by the sensory division of the PNS, or these commands going out to effectors of the PNS, have to travel through a series of neurons. The sensory information travels through about 3 neurons, one neuron towards the CNS, and then from the CNS on, there will be a couple more neurons before the information gets to its final destination. So there's pathways of neurons that take information to the CNS. Then the CNS, when it puts together a response, the response will go out in at least two neurons. So one neuron that takes it from the brain all the way down towards, out of the spinal cord, towards the muscle where it needs to go through another neuron. So there will be at least two neuron pathway, if not more. So there's these pathways of neurons processing or taking information back and forth. This is what we call the neuronal pools or circuits. Some of these circuits are diverging, in which one fiber stimulates more fibers, and the next round even more fibers, amplifying the information and going through several different parts of the body. So, for example, visual information sent to the visual cortex is being sent initially by one neuron. And when I say one neuron, one pathway of neurons, I should say, it's not one neuron, but one pathway of neurons. And from there, the information will begin to spread to other pathways of neurons going to different parts of the brain. So if this was a visual information coming in, you could even say through one nerve, eventually as it goes into the brain, it will be divided into or given to progressively more and more neurons going to different parts of the brain. So perhaps most of this information, this is visual, it's going to go to the visual part of the brain, the occipital cortex, but some of it may go to the cerebellum, which has to do with equilibrium, and it needs visual information. Some of it may go to the part of the brain that deals with visual reflexes, and they also need this information. So this will be a divergent pathway. A convergent circuit is exactly the opposite. Now you have several neurons that stimulate a single neuron. So this is the opposite. Again, this is common in sensory and motor systems. For example, the pathway that stimulates the breathing muscles can receive input from the cerebrum, because we could voluntarily initiate or stop breathing. Typically, though, it's coming from the medulla and the pons, in which we have signal for the automatic breathing, but it could also come from the emotional part of the brain. If we're upset, for example, that will also alter our breathing. So that pathway of neurons that are going towards the breathing muscles could be affected or have received input from several different pathways of neurons. A reverberated and oscillated circuit. This is neurons that contain collaterals that synapse with previous neurons, creating a loop. It's like a circular pathway that ends up, once it gets going, it keeps itself going. So we have the input going from one neuron to the next, but some of the neurons have branches that stimulate a third neuron, which in turn stimulates the previous neurons, and this pathway keeps going. Okay, so a reverberating circuit is sometimes used to maintain reflexes, for example. All right, so the serial processing is a stepwise processing in which one neuron gives information to the next, and that one to the next, and is more of a one step after another type of pathway, not converging or diverging, but it is more systematic. So this is a very simple type of processing, and that's why we present it. So this is a reflex, which is a very simple processing mechanism. So essentially you have information coming in from a receptor that's receiving a stimulus, and it stimulates it. It gives the information to a sensory neuron. Notice that the sensory neuron is a unipolar neuron, which brings that information to the central nervous system, in this case, the spinal cord. Notice that the information is not going to the brain, it's just going to stay within the spinal cord, at least for what we can see here. The spinal cord, in turn, gives the information to an association neuron or interneuron, which stimulates a motor neuron that goes out and affects a effector organ. So this is serial processing, and this is a reflex. Okay. And pick up reflexes on the next chapter. So I'm going to go ahead and keep going. If there are questions, go ahead and ask. Okay, so we're going to go over now the spinal cord, which is the last chapter, module 4. I'm not going to spend too much time on anatomy because this is part of the practical, so you should probably have already seen these. Now, functions of the spinal cord. The spinal cord is a way of communicating with the brain. So there are nerves that travel from brain on the spinal cord all the way, and then from there out through Pns nerves to an effector organ. Or there's information coming from receptors towards the spinal cord, and then the information is passed on a series of neurons that finally take the information, the sensory information, to the brain. So it's a two-way communication to and from the brain. This is also a side of the spinal reflexes, which is integration. So what that means is that some responses are not coming from the brain, some responses are coming directly from the spinal cord. So this is a spinal cord control of responses, and this is automatic. All right, make sure that you know the end of the spinal cord, the name of the end, and where it ends. The areas of the spinal cord: cervical, thoracic, and lumbar. Again, it ends at the lumbar area around L12. And then from L12, from the conus medullaris, the spinal cord proceeds with bundles of nerves called cauda-equina. You should know what a cauda-equina is also. The nerves that the PNS, this is going to be PNS, L12. Yeah, is it L12? Is that what I said? That should be it. Did I say L12? I meant L2. No, L2. That's where it ends. But it continues on from there as the cauda-equina. Yeah, I might have said L12. I meant L2. Yeah, there's no such thing as an L12. It's just 5 vertebrae in the lumbar area. Yeah, so the nerves that come out of the spinal cord are going to be PNS nerves, and they're going to be spinal nerves. I'm going to say come out, that would be motor nerves, or come in as sensory nerves. There are 31 pairs of spinal nerves coming in and out of the spinal cord. And we'll go on to what their names are here in just a second, or where they labeled. Okay. So the ones coming out of the spinal area are called cervical spinal nerves, thoracic area is thoracic spinal nerves, lumbar spinal nerves, and sacral spinal nerves. Enlargements are places where the spinal cord enlarges, becomes thicker, and that is due to the area receiving nerves from the upper limbs or lower limbs. So that is the cervical enlargement and the lumbosacral enlargement. Protections of the spinal cord, this is an important area. You probably saw these in lab. We're going to go ahead and cover it in lecture also. So the spinal cord is going to be protected by a series of membranes called meninges. Obviously, it is housed within a bone, so the bone itself, the vertebra, will be a protection. So we have the bone, then we have meninges, and then between meninges and actually spinal cord, there's also fluid that protects the organ. All right, so this is an actual model from lab, and you can see here this spinal cord right there. And this is the bone here. So notice that there is a space between bone and spinal cord. Okay, so they're not adjacent to each other. Okay, I noted these because we're also going to see meninges in the brain, and in the brain there will be no space. There will be an actual physical touching of meninges and bone. So this is the bone here. This is the first meninges right there. Let me go ahead and change the color here. So this is the first meninges right there, the dura mater. Okay. So there is a space between bone and the meninges, the first meninges, the outermost meninges. That space is called the epidural space. Second. Okay. Okay, I thought I had the epidural space in here somewhere. I don't see it, that's okay. No, it's another part. Oh, no, epinurium. No. Okay. We have it in somewhere else, but that's okay. All right, so that's bone again. It's there, I'm just missing it. It probably is. Okay. I do want to point something else out before I move on from this PowerPoint. So this is the first layer, which is the dura mater. And this is dura mater means tough. Actually, mater is mama, so it's tough mother. But this very tough layer, tough meninges, continues on past the spinal cord and goes out of the spinal cord and continues covering nerves. This yellow thing here is the nerves. So it becomes part of the PNS. It starts as covering the CNS, but goes on and begins to cover the PNS also. At that point, however, we're not going to call it meninges. We're going to call it epinurium. So we're going to change the name when the membrane leaves the central nervous system and begins covering nerves. It's essentially the same structure. We just changed the name because the location has changed. Okay, so I want to make sure I point that out, that the dura mater ends up becoming the epinurium. Yeah, we call it epidural space. Okay, so there's the epidural space. And again, this is something that is not going to be found in the brain. This is the place where an epidural injection will be delivered in between vertebra. And notice that it's not going to pierce the meninges. It's going to stay in the space above the meninges. Dura mater ends at about S2, so it goes past the spinal cord. So it continues going up until about here. That it goes beyond the spinal cord itself, which ends at about L2. This is showing you the epidural, which again, it goes in between vertebrae. It goes through the intervertebral discs into the epidural space. It should not touch the meninges, and it will deliver an anesthetic. Okay. I like this picture because it's very, very simple. And so here, these pink line is going to be the dura mater. Notice that the next meninges is the arachnoid mater, and it is touching the dura mater. So the two of them are adjacent to each other. Again, that's going to be another difference between brain and spinal cord, in which these two layers are going to be separated from each other. All right, so we have dura mater, arachnoid mater. Arachnoid mater is called arachnoid because it has these fibers that come out of it into the space called subarachnoid space, filled with CSF, with cerebrospinal fluid. And then the last meninges is the pia mater, which is touching the spinal cord. So arachnoid mater is touching the dura mater, deep to the dura mater. Below it is the subarachnoid space, which is filled with CSF. So in this picture, this space right here is the subarachnoid space. You can see that the give you a better view here. Okay, this is a close-up view. You can see here one line, that's the dura mater, and then next to it is another one, that's the arachnoid mater. Then you have this space, which is the subarachnoid space, where the CSF is found, one of the places where CSF is found. And then the last layer is one actually touching the spinal cord, which is the pia mater. All right. Okay, from the pia mater, there are these ligaments that come out called lenticular ligaments that are going to anchor the spinal cord. There's another one here, and there's actually two over here. Anchor the spinal cord to the dura mater and prevent it from moving sideways or up or front and back. So it is stabilizing the spinal cord, these denticular ligaments of the pia. The pia mater also keeps going. So this is the spinal cord right here. That's where it ends at about L2. Covering it is, not touching it, is the pia mater. And the pia mater continues on all the way down to the coccyx, to the little bone, and attaches to the coccyx. And that again anchors the spinal cord so that it will not move. So this little ligament of pia mater that attaches to the coccyx is called the filum terminale. Don't confuse filum terminale with conus medullaris. The end of the spinal cord is the conus medullaris. Filum terminale is the ligament that proceeds from the conus medullaris all the way down to the coccyx. All right, contrasting a epidural with a lumbar puncture. A lumbar puncture is a procedure that actually punctures the spinal cord, the meninges, going all the way into the subarachnoid space to retrieve cerebrospinal fluid. This is a completely different procedure, obviously much more invasive, in which in this case is not to deliver something but to take something away. So it's typically done in the lumbar area where the intervertebral discs are bigger and there's less chance of actually damaging the spinal cord. So the needle will go through the intervertebral disc, pass the epidural space, pierce the dura mater, pierce the arachnoid mater, and goes into the subarachnoid space where it will retrieve CSF. Yes, it's the same thing, spinal tap. Correct, same thing. Spinal tap and lumbar puncture are the same thing. Yeah. So the idea is to get CSF. Is it really? Okay. I would imagine it's painful. Imagine they only might give you local anesthesia, but it's probably very, very painful. Mm-hmm. Typically, when they want a CSF sample, is because they're looking for something serious, like maybe meningitis or some form of infection of the central nervous system. Okay. A lot of these is anatomy. So hopefully this is review. definitely expect in the test to have to identify structures from a cross-section of the spinal cord. The cross-section will be coming from the picture itself will be coming from the worksheets, PowerPoints perhaps, but what I have access to are worksheets and PowerPoints. So that's what these figures are going to come from. So make sure you take a look at the figures that are present in these resources, so that when you see it in the test, you know what we're talking about. You can identify the parts. All right. So first thing is to know what is front, what's anterior, what's posterior, and the gray matter of the spinal cord is this butterfly-looking structure in which the tips of the wing are going to be posterior and the more rounded are going to be anterior. So anterior or ventral versus posterior or dorsal. The white matter is surrounding the gray matter, and it folds into a sulcus posteriorly, making the posterior medial sulcus. And anteriorly, it falls into a deeper fold, more pronounced separation, called a fissure. This is anterior medial fissure. In the middle of the gray matter, there's a little canal called the central canal. And again, this will be another place where we're going to find CSF. Remember, white matter versus gray matter. Yeah, the little gray matter bridge that connects right and left gray matter, this little bridge right here, is the gray commissure. The white matter areas are called columns or funiculi. So there will be the dorsal columns or funiculi, lateral columns or funiculi, anterior columns or funiculi. Gray matter, okay, look at that. Gray matter are called horns. And so we're going to have the posterior gray horns, lateral gray horns, and anterior gray horns. Notice that the lateral gray horns are present in areas of the spinal cord with autonomic nervous system function. So the lateral gray horns are not going to be present in all of the areas of the spinal cord. Spinal cord and all central nervous system is highly organized, so that the posterior area is going to be sensory. The lateral and anterior areas are going to be motor. So sensory information is going to come into the spinal cord via the posterior roots. So these are going to be the roots coming in and arrive at the posterior gray horns. And these are going to be unipolar neurons. So this is going to be lots of little bodies of neurons outside of the spinal cord gathered together in one area, bringing their sensory information into the spinal cord via posterior roots and delivering it to neurons, bodies of neurons found in the posterior gray horn. Okay. This is even further organized because the somatic sensory is most posterior, visceral sensory is more anterior. Motor information or motor commands are going to go out of the lateral and the anterior gray horns. In this case, we have multipolar neurons of the A and S cluster in the lateral gray horns, and their axons will leave from the lateral gray horns through the making up the motor roots. Somatic motor neurons are going to be clustered in the anterior gray horn, and again, their axons will lead through the anterior roots. So lateral gray horns are autonomic sympathetic motor neurons, and they're found in the thoracic and lumbar regions because that's where sympathetic neurons are found. Anterior gray horns are somatic motor neurons, and they're found throughout the spinal cord. And again, this is showing you the same information: sensory from the back, visceral from the posterior gray horns, and motor is leaving from the lateral gray horns and anterior gray horn. This is little blue neuron here is showing you the sensory information coming in. Here's a little, these are unipolar neurons. So notice that all of the little bodies of these unipolar neurons bring in sensory information, be it visceral sensory or motor or a somatic sensory, are all clustered together. And any place in the PNS where we find gray matter, which gray matter is made of bodies of neurons, we call that a ganglia. So here's a ganglia right here. This ganglia is called the dorsal root ganglia. Ganglion for singular. So dorsal root ganglion, but there's a chain, there's lots of them all through the spinal cord. And here's another one from the other side. So you should be able to identify the dorsal root ganglia, the sensory or posterior roots, anterior or motor roots. And that's the same thing over here. Okay, more of the same. And this is showing you integration in the spinal cord. So you can see a sensory neuron coming in, bringing the information. There's the little dorsal root ganglia where the body of the sensory neuron is located. And it is giving the information to an interneuron, which in turn stimulates a somatic motor neuron, which takes out the command. Now, we talked about spinal nerves. A spinal nerve is actually not very long. It's a very short nerve that comes about when the sensory roots coming in and the motor roots going out come together and travel together under the same wrapping, the wrapping here being essentially making up the nerve, being a layer of epineurium wrapping around all of these little nerve fibers going in and out. And this is just long enough to leave the from in between vertebra. So that's what we call a spinal nerve. This picture, this model is very good because it gives you a good spinal nerve. So here you have the little spinal nerve right there, which it consists of both sensory and motor fibers. And so any nerve that has sensory and motor fibers, we call it mixed nerve, versus something that is only sensory or only motor. So spinal nerves, all of them are mixed nerves. This will be the sensory roots. So here are the sensory roots. There's the little dorsal root ganglion, motor roots down here. Anyway, so be able to identify those structures. These structures are not part of the spinal cord, but they're associated with the spinal cord. So we'll go ahead and cover them at the same time. Spinal reflexes, we talked about reflexes already. Reflexes is a very simple integration example. That's what we study them, is because this is the simplest integration. It happens at the level of the spinal cord, at least the ones that we study. Not all reflexes are just spinal cord, but many are spinal cord reflexes. So this is very, very simple information coming in, command going out. All right, so the basics of a reflex. This is what we call a reflex arch. Sensory information coming in into a sensory receptor. The information travels through the sensory nerve, reaches the central nervous system, which will be the integration, and the sensory nerve gives the information to an interneuron. Interneuron synapses with a motor neuron, which leaves the spinal cord towards an effector. So there are 5 components in a reflex arch. Actually, there could be as few as 4, because this middleman right here could be in some instances eliminated, and then we have a direct connection between sensory and motor neurons. So the minimum that a reflex arch will have is 4 components: the sensory neuron, or I guess you should say the sensory receptor, the sensory neuron, motor neuron, and effector organ. Okay, that's the minimum. Most of the time, however, we do see an interneuron. The more components you have, the slower the reflex, because each component represents a delay. It's like having runners. You can have 5 runners, or you can actually have as few as 4 runners. But every time you give the information to the next runner, there's a delay. So that makes the whole process a little bit slower. So the fastest of reflexes are the ones that do not have an interneuron. Okay, so again, that's just showing you the reflex and the components. So a reflex is an automatic response to a stimulus produced by a reflex arch. There are many ways to classify reflexes. The only way they're classified in this chapter is monosynaptic versus polysynaptic, meaning one synapse, sensory to motor, no interneurons, or polysynaptic with an interneuron in between sensory and motor. This is an example of a monosynaptic reflex. Yeah, this is a patellar reflex, which happens when you go to the doctor and a hammer taps on the patellar tendon. And essentially what that's going to do is it's going to pull on the tendon of the quadriceps muscle. The proprioceptors wrapped around the muscle fibers are going to sense the pull and send that sensory information to the spinal cord. This is a monosynaptic reflex because the little sensory neuron is directly connected to the motor neuron, and the command goes out to contract the muscle. See, the stimulus is stretching the muscle. The response will be to do exactly the opposite of what the muscle is doing to prevent overstretching. If the muscle stretches, the reflex is going to contract it. If the muscle is contracted, the reflex will stretch it because it is a gear to prevent overstretching or overcontraction. So the response will be to contract the muscle is contracted, the leg goes up. Okay. So that's the knee jerk or patellar reflex. It's a monosynaptic reflex. A withdrawal reflex, so extension reflex, is a polysynaptic reflex. And some of them can get pretty complicated. This one right here is showing you somebody stepping on a tack, and now you have 2 things going on. You have one leg that is going to have to flex, while the other leg is going to have to extend in order to catch balance. So the leg that is getting the stimulus goes into the spinal cord, connects with a interneuron, and this is a polysynaptic reflex. The motor neuron goes out, and in this case is innervating the hamstring muscles, and the leg is going to flex, moving away from the source of pain. At the same time, that sensory neuron is also stimulating another interneuron that's going to cross the other side of the spinal cord and stimulate a motor neuron for the other leg to extend the quadriceps muscle, extensor muscles, to extend the leg, the other leg, and catch balance so the person doesn't fall. So you have several connections here. Then the sensory neuron also goes up and informs the brain of what's going on. And that always happens. The brain is always informed of what's happening, and so it's aware of the reflex. And the brain could stop the reflex if it wanted to, actually. All right, so that's the polysynaptic reflex. So know the difference between a monosynaptic and a polysynaptic reflex. Back to spinal nerves and how they're organized, very similar to the way that skeletal muscles are organized. The neurons in the nerve are going to be covered by a connective tissue called the endoneurium. So the axon of the neuron is covered by the connective tissue, each axon of the neuron. Many of these axons are bundled together into a nerve fascia by a connective tissue called perineurium, and then many fascicles are bundled together by epineurium. So it is exactly the same organization as skeletal muscles. Dermatomes. Okay, so spinal nerves. Let me come back to dermatomes in a second. We mentioned that spinal nerves are nerves that leave from the spinal cord. From each region of the spinal cord, we have a series of nerves, and the way they're going to be named are going to be according to the region of the spinal cord from which they are coming in or out, as to say what they're associated with. Okay, so let's explain how that works. All right, so here you have the cervical region of the spinal cord. And you have vertebra number 1, C1, C2, C3, C4, C5, C6, 7. The cervical region has 7 vertebrae. Okay, so let me go ahead and draw them here because it's a little bit hard to see on the picture. So that's C1, C2, C3, C4. Now I wish I made him smaller. C5, C6, C7. Then I want to make T1. I want to make C7 and T1 over here because those are important ones. C7, T1, might as well make C6 and T2. Okay, so skull right here. Maybe not. Okay, not wanting to work. Let's change colors. Nope. Okay, there we go. So that's the skull right there. So the first cranial nerve is going to come out between the skull and vertebra C1. And we call that spinal nerve C1. Good name for it. So that's spinal nerve C1. Second spinal nerves come out from C1 and C2, and we're going to call it C2. Okay, that's pretty easy. Next one comes out between C2 and C3, and we're going to name it C3. So these are the names of the spinal nerves. Between C3 and C4 comes out C4. Between C4 and C5, C5. Between C5 and C6, C6. Between C6 and C7, C7. Okay, so we have C7 right there. And this is what they did next. Between C7 and T1, instead of calling it T1, they called it C8. There's no such thing as a cervical vertebra C8, but there is a spinal nerve C8. And part of the reason why they did that is because C8 kind of goes to the same places as the other cervical nerves. So it made more sense to put it with the cervical nerves than spinal nerves than with the thoracic ones. Now we come to the thoracic spinal nerves. Between T1 and T2 comes T1 and then proceeds on from there. So there's going to be 12 thoracic spinal nerves, 5 lumbar spinal nerves, a couple of sacrals. Okay. You count them all, you should end up with 31. What should you know? You should definitely know that spinal nerves in the cervical region are named after the vertebra below. starting with the thoracic region, spinal nerves are named after the vertebra above. So T9, you should know, is the spinal nerve found between vertebra T9 and T10. You should know that spinal nerve C4 is found between vertebra C4 and C3. So when the question is, which spinal nerve comes out of vertebra C4 and C5, the answer is C5, the spinal nerve C5. Okay. So that's basically how we name spinal nerves. Also, we have mapped where these spinal nerves go to, what regions of the body they serve. These regions are called dermatomes. So you can see here the region for C4 right there, and for C5 right here. This is anterior versus posterior over here. Okay. And the dermatomes, this mapping helps if there is nerve damage. There may be, for example, loss of sensory feeling on the C4 region, and that tells us that maybe nerve C4 has been damaged. So it allows us to map the regions that these nerves serve, and if there is a damage to the nerve, there may be some abnormalities in those regions. Now, the regions are not as clear-cut as they are in the pictures. There is some overlap of nerves, but it still works. All right, so you should know what dermatomes are. And again, you should know what are the names of the spinal nerves. Okay, we have seen these already. Now, spinal nerves are not very long. Very shortly after they leave from in between vertebra, they begin to branch. And the branches are called rami, right there. And so this right here is the spinal nerve, right there. And notice how it branches into a dorsal area, dorsal ramus, going to the back, ventral ramus, going towards the front, and then there's these rami communicants, two of them, that are going to actually go into a ganglia called the sympathetic ganglion. And these two together are called the rami communicants. All right, so dorsal ramus serves the back, the trunk, the posterior aspect of the trunk, the back. So all of the muscles and skin of the back. These are all mixed branches, so this is mixed, taking in sensory and motor fibers. Ventral ramus is going to serve the anterior and lateral aspect of the trunk plus arms and legs. Again, this is mixed. So if you have a person now, you have taken care of the back of the trunk and the front of the trunk and the lateral aspect of the trunk and the arms and legs. What you have not taken care of is the viscera, all the organs in the thoracic and abdominal pelvic area. The organs are not being, we don't have, as far as what we've seen, nothing goes back and forth to the organs. All right. Most of the sensory aspect of the organs, meaning interoceptors, are going to be served by cranial nerves. When it comes to motor commands going to organs, these are going, the ones that are carried out by spinal nerves are going to be sympathetic. Okay, so sympathetic motor going to organs are going to travel through the rami communicants. So the rami communicants are not mixed branches. These are motor branches. Only ANS fibers, which are motor, travel through it, and even more specifically, only sympathetic motor fibers travel through it. Okay. When it comes to parasympathetic, most of the motors are going to be cranial also. So that's why we don't see sympathetic here, or parasympathetic, I should say, is because we're dealing with, these are spinal nerves, not cranial nerves. All right, so you should know the branches and where they're going to and fro. In the picture, this is the dorsal ramus here. This is the ventral, which is huge, and you can see one of the rami communicans, the other has been cut. Okay, and last, we're going to look at some of the spinal nerves themselves. So from the spinal nerve comes the rami communicans, the ventral, the dorsal ramus. From there, they're going to branch even more, creating these networks of nerves, which we call plexus. And so there's a cervical plexus, a brachial plexus, there is a lumbar plexus, sacral plexus, and coccygeal plexus. The only place where there isn't this network of nerves combining with each other, but rather spinal nerves leave very cleanly from there, in between ribs, is the thoracic region. So there's not a thoracic plexus. From the cervical plexus, the main nerve you need to know is the phrenic nerve, which is important because it innervates the diaphragm, which is a breathing muscle. In the brachial plexus, we have the axillary nerve under the arm, which innervates muscles of the shoulder, like the teres and deltoid. And obviously, since the skin of the shoulder is being, sensory information from the skin of the shoulder is being brought back by the axillary nerve. Radial nerve is going to innervate some of the flexors of the upper limb and brings back sensory information from about a third of the hand, the surface of the arm, and forearm. Musculocutaneous is another one of the brachial plexus. So the brachial plexus has several nerves that we need to know: the axillary, radial, musculocutaneous, and ulnar. So musculocutaneous and ulnar nerve. And the median, I forgot about the median too. Yeah. So we have axillary, musculocutaneous, radial, ulnar, and median. So 5 nerves from the brachial plexus, all of them serving the upper limb. Sacral plexus, obturator nerve serves all of the adductors muscles, the inner thigh muscles, is this one right here. And femoral nerve is huge, and that one is going to serve the anterior and lateral aspect of the thigh, the muscles like the quadriceps muscles, iliopsoas, sartorius. The femoral nerve begins to travel towards the medial aspect of the thigh, and then it branches into this long nerve called the saphenous nerve. So the saphenous is a branch of the femoral. This is the knee here, this is the leg. So the nerve is coming from the thigh on the medial aspect. So you can see here the saphenous is this long nerve right there. So these are lumbar plexus nerves: the obturator, the femoral, the saphenous. On the sacral plexus, we have the biggest nerve in the body, which is the sciatic nerve, this huge thing here that travels posterior aspect of the thigh. And that's why in this model, you can see the foot rotated to one side, so we can see the posterior aspect of the thigh, this huge nerve called the sciatic. At the level of the knee, the sciatic splits into tibial and common fibular. And actually, this is one of the branches of this split. The other one is going to go to the front. Okay, so this is what we were looking at. This right here is this next picture right here. Okay, so this is sciatic, and this is the split right there. So this is split going towards the front aspect of the thigh, the lateral aspect of the thigh, is the common fibular, because it's going lateral. The fibular bone is lateral, and the one that stays on the back is going to be the tibial, which later is going to split into two more, but I don't think we'll go into that. The common fibular, now we're looking at the other leg. Now we're looking on this side right here, on that leg. Oh. So the common fibular, again, is a split of the sciatic, goes to the front on the lateral aspect, and goes down as the deep fibular and superficial fibular. Okay. So you should know these nerves, spinal nerves, and have an idea of the areas where they serve. These are mixed nerves, so they're going to have sensory and motor functions. Okay. Questions?